Okay, thank you very much. I want to thank the conference organizers for inviting me here to give a talk. It's been a very interesting meeting, and I often don't get to speak to so many people with similar interests, so it's a real treat. I'm from the University of Vermont, and I've listed some of the people that are involved in this talk. And what I'm going to talk about today is, can evolutionary change lead to ecosystem transformation, a model system approach? And what I mean by that is I'm going to take both a model biological approach and also apply some theoretical models to try to ask, are we changing evolutionary processes so much that it's going to lead to ecosystem transformation? Okay, so this is just my introductory slide where setting up the idea that there are eco-evolutionary dynamics. In this particular audience, I probably don't need this slide. We're all aware from this meeting that genes can influence phenotypes, can influence populations, communities, and ecosystems, and there can be feedbacks between different levels of organization. But what I'm particularly interested in is the idea that some of these feedback relationships can be changed, and they can lead to what I'm calling what's called a regime shift. So we can go from a state where we have a natural wetland, which is, I study wetlands, so we can go to a native ecosystem where we have a natural wetland, and through a change in feedback relationships, we can undergo a critical transition to an invaded wetland that has different ecological properties than the original system. And so the question I'm asking is, and we can get then a complete and total ecosystem transformation. And this is just showing a wetland completely invaded with the grass I study, which is Phalaris arundinaceae, which is reed canary grass. Occurs pretty extensively in Europe, but it's very invasive in the US, especially in the Midwest where they have high levels of eutrophication. So the question I'm going to talk about today is, when do these invasive plants lead to regime shifts? And are evolutionary processes driving these regime shifts? Can they be driving these regime shifts? And there are several general properties of regime shifts. And I'm going to use the definition that's of Norberg and Cummings, where they say that the ability of a system to internally switch between different self-reinforcing processes that dominate how the system functions. So when we get a series of switches in the feedback relationships, we can get a transition from a system which has a lot of negative feedbacks to a system that has a lot of positive feedbacks. And when we get a switch in those kind of feedback relationships, we can undergo a critical transition to a new state. And once we've undergone that critical transition to a new state, we actually requires active management to get back to the original ecosystem. So it's no longer enough just to remove the invasive species. We actually have to do active management. So try to avoid that. And can evolutionary dynamics in invasive species actually lead to these regime shifts? And the reason that I think that they might is because there can be new traits or new trait combinations that arise in the new range that weren't present in the old range, and these could lead to new functional traits that have different effects on ecosystem performance. And these new traits can weaken or reverse ecological feedbacks that lead to an ecosystem transformation. To do this kind of work, it's important to take a biogeographical approach when you study invasive species. So it's important to actually know what the traits are in the native range and how they're actually changed in the invasive range. And there's a lot of biological invasion literature that does that. And it's also important to know that invasive plant populations are different from their native counterparts when you study the same species. And this is the work of Blumenthal and Hufbauer, where they studied 14 different invasive populations and their native population counterparts in common gardens, and showed that, in fact, many of these invasive populations were larger than the native ones in a common garden. And this is a very common result, that invasive plants grow larger in the new range than they do in the old range. And another common result in invasion biology is that there are multiple introduction events. And so one of the things I'm interested in is, how do these multiple introduction events translate into changes in evolutionary processes? And so there's several studies that this idea that multiple introductions are important in invasion is becoming more and more apparent. There's the work of Colby et al. Studying lizards, my own work studying reed canary grass, the work of Cully and Hardiman studying invasive pear tree in North America, and the work of a colleague of mine, Kaylor and Taylor, studying an annual Silene plant. And those are not all of the studies that have shown that these multiple introductions are important. But when we have multiple introductions, we may be changing the evolutionary processes. So one thing that happens when we have multiple introductions is we're taking individuals from different parts of the native range and we're combining them in new ways. And when we do that, we're getting admixed populations. And I've heard a little bit here about admixed populations. And one thing that can happen when you get admixture is that you can get traits expressed outside of the parental type. So what I'm showing here, is the idea that you have species A, which is little sunflower, and species B, which is a big sunflower. And you see that when we mix them together and make the F1s, we get some that are very small and some that are much bigger than the parental type. And the other diagram showing that, so when we look at the mean, well, that's not helpful. Sorry. When we look at the phenotype, you see that there's different population peaks of the parents, and there's some individuals that can end up way on the tail. And according to Riesberg, those are called transgressive hybridization events, when we see individuals that are far beyond the trait values of either of the parental populations. And so I'm going to start with this conceptual model where we have admixture due to multiple introductions. And that admixture is creating new functional traits. And those functional traits are translating into different whole plant traits, and that's causing a community response. And we get to the next spot that may be changing key ecosystem features, changing known feedback relationships and reinforcing features, such that we can get to a new ecosystem state. And I'm going to tell you now about the biological model and some data I have that goes along with this. And when you speak so late in a session, I was deciding whether I should present the biological model first or the mathematical model. And I thought that maybe I'd present the data first and then just kind of glance through the model. So I'm going to present you some of the data we have that supports some of these ideas and actually led to the development of these ideas in my lab. So the biological model is the plant I've been studying, which is reed canary grass. It occurs widely in US and Europe. It's a perennial grass. It reproduces both sexually and asexually. It has an outcrossing mating system. And it's been introduced multiple times from the native range. It's for pasture forage, for ditch stabilization. It comes in inadvertently in seed mixtures. So it's been introduced multiple times, and we have evidence for that. And I took a biogeographical approach where I collected from both Europe and the US. And when you go to study these plants, you get to go to very interesting places, but the wetlands always look about the same. So I decided I'd present actually the locations where I collected the plants. So I collected the plants from near Trebon, Czech Republic, which is, I was a guest at the Institute of Plant Ecology, and also in southern France, in Montpellier, where I went to the Camargue and collected some plants. I then collected in my hometown, which is Burlington, Vermont, that's Church Street, and I collected in the Smoky Mountains, which is the southern extent of the range in the southern US in the Smoky Mountains. And some early work we did, we showed that there were multiple introduction events and that they were resulting in higher genotypic diversity. So both of the invasive populations groups, the Vermont and North Carolina ones, had more total genotypes present than either of the native range ones. And that in addition, there was less of a reduction in genotypic diversity at the range margin in North Carolina, the invasive range, than in the native range in France, where they see a large reduction in genotypic diversity. And we also showed that these multiple introductions were leading to new gene combinations, and that there were certain private alleles that were present in both the Czech Republic and France that co-occurred in Vermont and North Carolina, and that although there were more genotypes present in the invasive range, very few common genotypes were coming over. And although we didn't sample extensively, it's unlikely that there was only 3 individuals that came over as known multi-locus genotypes, just in that there's a lot of mixing that's going on in this system. And how does this then change key traits? Well, this was kind of a surprise to us, because what we found when we looked at genome size, we found that there was a reduction in the genome size in the invasive populations when compared to the native ones. And this is quite an interesting result. And we wondered, because we hadn't sampled very extensively, whether this was a general result or not. But more recently, Laura Meyerson, who did an extensive sample of both the native range and the invasive range of a similar species, Phragmites, which is also invasive, found that exactly what we found, that the invasive populations have, on average, smaller genomes than the native ones. And interestingly, this smaller genome size in the invasive populations is correlated with a faster stem elongation rate. And what's interesting about that, to me, is that when you look at the white circles, which the open circles are the native ones, and the black ones are the invasive ones, and they overlap in their distribution. But when you look at the native genotypes, there's no relationship between genome size and growth rate. But when you look at the invasive ones, there's a clear relationship between smaller genome sizes having faster elongation rates. And that led us, and so that was an interesting trait that we wanted to investigate more. And we took, because I collected these individuals from different locations in the field, and because they're a grass and they clonally replicate and you can put them in your suitcase and bring them back, I do have phytosanitary permits. So we could take a phytometer approach and bring the individuals back and do a series of experiments on the same individuals under many different conditions. And we've done that, and I'm only going to present some of the work we've done. So we dig up the plants in the field, they can be cloned, and then we do experiments on them in different conditions. And one of the things that we were able to do when we grew the plants in a common garden is we looked at the genetic architecture of the plants from the native range and the invasive range for 3 different traits. And these are emergence time, height, and tiller number, 3 things that are maybe correlated with fitness. And it looks very nice in that the invasive ones look different than the native ones. But when you break that down and look at the architecture, the genetic architecture or the G matrix between the different regions, you can see that it looks like something different is going on, which maybe isn't surprising, but it looks like the Vermont ones have been released maybe from some kind of genetic constraints, and the French ones and the North Carolina ones have a small range of, or in the small range of the space, but they look like they're doing slightly different things. We also looked at the phenotypic plasticity that was present in these populations, with the idea that there's an idea that invasive genotypes would be more phenotypically plastic than native ones, and that perhaps they were using a different strategy. In this case, this is showing the work of Richards et al., done a few years ago, where she proposed that invasive individuals in a species may actually show new strategies, and what she called one Jack and Master, in that it was both better under favorable conditions and also didn't decline as much under stressful conditions. And we have some data on that for our invasive and native populations, where we grew the plants along a moisture gradient, and I was calling the ones when they're sitting in moisture stressful, although it doesn't appear to be stressful for the invasive population. So you can see that for the native genotypes, they're stressed by water when they're sitting in water, but the invasive ones actually show an increase. And that the top one is for biomass, the second, I believe, is for height, and the third one is for tiller number. And even when they have the same or almost overlapping in the favorable environment, when they go to a stressful, which may not be stressful for them, but a water environment, you always see that the invasive ones are doing better. Getting back to the main story, though, are there any other traits that may be altered that would be important and may change feedback relationships? We looked at another trait, almost anecdotally, which is carbon to nitrogen ratio of the leaves. So what we found is that the invasive populations have a higher carbon to nitrogen ratio than their native counterparts. And in addition, the invasive populations have a faster growth rate than the native counterparts. And this makes sense because it's basically the plants are growing larger, and there could be a dilution of the nitrogen effect, resulting in the fact that the carbon to nitrogen ratio in the invasive ones is higher than in the native ones. And that could actually change feedback relationships. If you look at the following diagram, this shows a cartoon of possibilities that may be happening. So what we have is a situation where we have the invasive populations are growing larger. they're having a greater carbon to nitrogen content in their leaves. And they're bigger, so they're leaving a bigger litter signature in the ecosystem. In addition, the higher carbon to nitrogen ratio present may be causing a slowing down of the decomposition rate, causing a further positive feedback relationship. And we have some evidence from my own lab that this higher carbon to nitrogen litter composition actually does slow decomposition, but it's a well-known result from other people as well. And so what happens is that we're making a prediction that this change in this carbon to nitrogen ratio and the fact that the plants are larger, we would expect that there would be actually a larger litter signature in invaded populations than native populations of reed canary grass. Oops, I think that's the wrong way. And so we wanted to ask, is there actually a signal of this in the ecosystem? And this is the work of my former postdoc, Martin Eppinger, where we actually went back to the Czech Republic, to 4 sites in the Czech Republic and 4 sites near my home in Burlington, Vermont, and we collected litter, 8 plots of small plots, 50 by 50 centimeter plots, and we collected all the above ground biomass of all species on it and then separated into above ground living biomass and dead biomass or litter. And when you look at the results are consistent with our prediction, and that is that the USA sites have significantly more litter than the Czech Republic sites. And even when we take out the outliers, the results are still statistically significant, although this is just simply a correlative result and just a thought experiment at this point. But why do we think that these changes in the carbon to nitrogen ratio, that's changing the amount of litter in the system, can alter feedback processes and cause an ecosystem transformation? Well, there are 2 things that litter feedbacks can do. One is it can alter the light reaching the soil surface. So as you increase the amount of litter that's present in the system, there's going to be less light available. And for the invasive plant, for Phalaris, it's a very good competitor for light. But litter also can alter the nutrient profile. So even if there's more litter, there's the decomposition rate is more, the litter persists for longer, it leaves a longer signature of nutrients in the soil, and Phalaris is actually a poorer competitor for nitrogen and nutrients. So those 2 situations set up a situation where positive feedbacks can be important in the system and drive the system to a new state. So what we did is we took some data that was done on this, some experimental data of Laura Perry and Susan Galadowicz working on a native sedge and in competition with Phalaris, where they actually looked at competition along under different light conditions and competition along different nutrient conditions. And we used that information to parameterize a resource ratio model. And what's shown here is the results of that model. And the 2 gradients are a light gradient and a nutrient gradient. And the first panel shows no litter feedback. This one shows the nutrient litter feedback, the light litter feedback, and both feedback relationships put into the model. It's a phase plot. So 1 is there's nobody can survive. 2 is the Phalaris only survives. 3 is coexistence. And 4 is the native only. And what you can see is just by putting the nutrient feedback in, you've increased the area over which Phalaris can outcompete the native species, and you've reduced the area of coexistence. When you look at the light litter feedback, you see a similar but not as dramatic result. But when you put both feedback relationships into the model, you see a very dramatic, you see there's still an area where you can get some native sedge existing, but most of the parameter space has only the Phalaris. And so you can ask yourself, well, how big a change in the carbon to nitrogen ratio, which changes the decomposition rate, would you need to see to cause a regime shift? And this is simulation data that we did, where we start with a system that has no invasive individuals at all, and then we just keep changing the invaders' carbon to nitrogen ratio in their leaves, and we look to see when we get a critical transition. And what this is showing is that there are these effects the system will stay in a native ecosystem state unless it's perturbed by a little bit of nutrient pulses, which is actually, in terms of the conference, this means that human alteration of the system by just adding a tiny bit more of nutrients could cause a shift of the system from a system where we had a native-only state to an altered state where the only thing that will survive will be the invasive phalaris. So getting back to the idea of admixture, so we were showing how the change in the mean trait value would affect these would affect these ecosystem transformation. But what's interesting to think about is, well, what happens when we are increasing the trait variance? And now we have a greater range of possible strategies that are available to the invasive genotypes than to the native genotypes, which is what I showed earlier. And the second question is, actually, do we have any evidence of these transgressive segregation in invasive genotypes? And this is showing some data I have on carbon to nitrogen ratio, which I'm calling a key functional trait. And what I want you to notice is that, in fact, there's great overlap between the carbon to nitrogen ratio in the native and invasive ones. But at the tail of the distribution, we're only finding the invasive populations. So maybe this is an example of transgressive segregation, I really don't know, but we can still model it. And this is actually a spatial model. It's a stochastic cellular automata model, and the way to read, there's 3 different panels, and the way to read the diagram is there's light availability on this axis and nutrient availability on the horizontal axis, and the panels are, the black is a native Carex species, and then there's competing with 10 invasive genotypes. In the first panel, it's only the carbon to nitrogen ratio found in the native genotypes and no litter feedbacks. In the middle panel, it's the Carex competing with native genotypes only with litter feedbacks. And in the third panel, we've actually put in what I'm calling the transgressive hybrids, or the segregation, and that's the native genotypes with the invasive genotypes with both litter feedbacks. And what you're seeing is that as we add in these more extreme genotypes, there's a greater range of the parameter space of the spatial location that they can occupy, and they're moving out the native sedge. And although what I presented today is a retrospective analysis, taking data I already have in my lab and using models to make predictions, what we're hoping to do is a prospective analysis where we actually can create these admixed individuals in the lab, find out actually does their genome size change in the way we've seen, and measure their trait expression under different conditions. Because we're dealing with a little grass, we can set up mesocosm experiments and look to see whether the feedback relationships that we're predicting to change actually occurs in these little experiments. So in summary, the ecoevolutionary dynamics of phalaris, I'm proposing that this admixture is creating a change in the functional traits, and these functional traits could be as simple as a change in the leaf carbon to nitrogen ratio and the total biomass of the plant. That's leaving a greater litter signature in the invaded ecosystem, and that greater litter signature is actually maybe important enough to create a critical transition to a new ecosystem state. Thank you very much. And I just want to acknowledge my, my colleagues and collaborators. These are all former associates of my lab. This is all done in my lab. My former postdoc, Sebastian Laverne, did the genetics work. My former postdoc, Martin Eppingate from the University of Utrecht, did the modeling work and some of the litter work. My former student, Matt Caproth, did the litter feedback work. And my collaborator, Stephen Keller, did the admixture work. So thank you very much for your attention.